So our next presentation will be given by James Borrell from the Kew Gardens in London, and I hand over the phone to you. Great. What's yours?
Thank you very much, and thank you very much. So thank you, everybody, and what a pleasure it is to talk at a room full of tree people. Normally, like, there's lots of zoologists and stuff at conferences, and, you know, they don't find it so interesting. So, yeah, what a pleasure. So I just start by saying a lot of this was done at Queen Mary, and I'm now at Kew, and working in partnership with some really good conservation organisations in Scotland. So I guarantee, I think the tree I'm going to talk to you about today is the smallest one that any of you study. On a really good day, if there's no deer, it might get to about waist height. So it is Dwarf Birch, the best birch. And why is it interesting and why is it important? So Dwarf Birch in the UK and in Scandinavia, where it's common, is a tree-line species. So after all the nice forestry trees can't grow anymore, and all the foresters have stopped walking up the hill, hopefully you get this nice transition of these sort of stunted trees that transition into your moorland. And unfortunately in Scotland, we more or less don't have any of that anymore. We just have forestry, forests, or we have moorland for grouse and deer and things like that, which is a shame because together with the mountain willows and the junipers, it's a super nice habitat. And we're realizing now that, of course, it can do things like slow down water runoff, reduce erosion, and all kinds of useful things. So I'm going to talk to you about Dwarf Birch in Scotland, because with our conservation partners, it's now something we're actually interested in restoring, as well as all these other beautiful trees. Because if you want to be able to claim that you've restored a huge area of thousands of hectares of continuous forest, in Scotland you have sort of 600-metre passes and things like that, and the only tree you can put up there that will let you claim that you've got a continuous canopy or forest are things like Dwarf Birch. So it's a really important part in sort of connecting up habitats over these vast areas in countries like Scotland. So why else is Dwarf Birch good to study? Well, from a conservation perspective, it's got almost every problem you can have, as well as being a wee tree that never escapes the pressure from deer. It also goes up in flames when you burn the moorland. It's got reduced connectivity like all of our other trees. And of course, it's a montane species, so climate change is a bit of a disaster. So it's kind of got everything wrong with it that you could have as a tree, which is really nice for people like me. We know that it probably moved southwards during the last glaciation and was one of the first trees probably back into the UK after Europe started to warm up. So it's got everything wrong, which is great. So how can we start mapping that? Well, we put together, people in the UK have been counting trees and mapping them for a long time, which is also very nice. And you can see, without any sort of statistical manipulation, that the population has been declining in Scotland and is now very, very fragmented. What we've also, I won't show the data today, but what we've also shown is we can detect the footprints of introgression in the other two British birch species further south. So we can see a little tiny bit of introgression into Dwarf Birch in southern UK, into Betula pendula, Betula pubescens, and that amount increases as you go further north until you actually find the real deal in Scotland. So this is what's happened to Dwarf Birch. So what are the three questions I'm going to try and talk to you about today? So before we can understand anything about what these processes have done, now we need to understand the past demographic history of Dwarf Birch. Second, a lot of us are conservationists, so we're interested in how you work out which ones to conserve, how much to conserve. And then in the future, I'm really pleased that Sally said that everyone in Canada really likes the idea of assisted gene flow. I wish if we did a survey of our reviewers in the UK, they had the same sort of 83% support for assisted gene flow. But what would assisted gene flow actually look like in the UK? And just to give you a flavor of the sampling we did, 30 populations in the UK. If you can just see the black dots here, these are populations we went to but couldn't find them anymore. Dwarf Birch is a little bit hard. It can sometimes hide below the grass. It's that small. But you can see definite pattern of it's the southern populations that we could no longer find. And then we also sampled in northern Scandinavia, which was super nice to go up to northern Scandinavia. You pretty much can't tread anywhere without treading on Dwarf Birch. So it's a really nice comparison of a not fragmented and a non-declining population. We did microsatellites, sort of conventional microsatellites, as well as RADseq. And then I also just want to talk very, very briefly, we developed a method to mine microsatellite markers from RADseq reads. And that's really nice because it gets rid of a lot of the biases that sort of those traditional wet lab microsatellites have, where you're looking for variability, you don't want to do too many, and you multiplex them. So what we ended up with was sort of real microsatellites, traditional PCR ones, RAD microsatellites, an independent data set, and then we divided our SNPs into transitions and transversions. So we ended up with 4 sort of semi-independent data sets with which to go forward. So the first thing we did is an ABC to look at the past population demography. I won't tell you all the scenarios we looked at. We spent months playing with scenarios. But the main one we were trying to differentiate between was a bottleneck before colonization of the UK, so before those Dwarf Birch came across Doggerland, or a bottleneck later, which you can see here, which after divergence of UK populations, something we did, maybe something like overgrazing, deforestation, things like that. And sure enough, we got a lot better support for this latter scenario where Dwarf Birch colonized the UK, diverged from Scandinavia, the populations diverged into all those hills and mountain ranges in Scotland, and then we had a very severe population bottleneck. We can see here some estimates of past effective population size in Scandinavia and the UK, and you probably can't see at the back the 4 sort of colored lines here, but each line is from one of our marker sets. And what you might be able to tell is that this line, the red one, has pretty much no signal. It tells you nothing, and that's the microsatellites. They tell you nothing about past events a long, long time ago because you've got lots of homoplazy now, they've kept mutating. But the SNPs were very good at telling us about these past events. And by combining all of these data sets together, we get really nice, clear signals. Then if we look at more recent events, incidentally, the microsatellites, if you're looking at something that happened recently, are much better. They're even better than SNPs. So I'd say, microsatellites aren't dead. I'm a big fan of microsatellites. But what we do see is that the divergence with Scandinavia, if we take an approximate generation time of 10 to 15 years, comes up about 8 to 12,000 years ago. So that kind of makes sense when we know plants would have started to recolonize the UK. In terms of a divergence in the UK, about 300 years ago. And again, that makes a lot of sense with when we shot the last wolf, when deer numbers started to explode, and Dwarf Birch would have stopped being able to have gene flow between populations. And then the most severe bottleneck was perhaps only about 10 years ago, 10 generations ago, about 100 to 150 years ago. And that's so recent that it almost becomes difficult to tell that it isn't just happening now as well. And that we attribute partly to those other things, but also to climate change as well. So we know something now about what's happened to Dwarf Birch in its past population history. So which ones should we conserve? As a conservation biologist, I'm always thinking the most unique ones, the ones perhaps on the southern range edge, the ones that look different from the other populations. We're trying to capture that diversity. Well, what this plot is this is a plot of maximum likelihood FST. We take each population for each marker, and then we compare it to the mean allele frequency of the whole meta-population, if you will. So how unique is each population? And what we actually find is that overall, the diversity in Scotland is relatively similar to Scandinavia. Everything's actually okay, even though we've got much smaller, more fragmented populations. They're very, very similar. The squares are Scandinavia and the circles are Scotland. And what we interpret that as meaning is that this fragmentation that's happened in the UK, this has isolated and partitioned the diversity. But when taken as a whole, it's still very similar to unfragmented Scandinavia. It's just partitioned into subunits that are starting to drift. So that's good news for restoration. Scotland still has all the diversity it needs, but it's just a bit of a ticking time bomb of genetic drift in all these subpopulations. Genetic drift only gets really bad when your census size goes down, and these populations start getting crazy FSTs of 0.6, even up to 0.8. So these populations are subject to really, really severe genetic drift, but the good news is it only happens when you get really, really, really small. So what is it like if we think to the future? So the first thing we did was a species distribution model for Dwarf Birch. Now, I'm sure, as you all know, anyone can put data into Maxent and get a species distribution model out. Doesn't mean it's any good. So what we did was we measured total catkin count and percentage germination, and we compared that to our habitat suitability values. And you cross your fingers and you close your eyes and you really hope. And what it shows is that places we say are good, so the light green there, places that are good habitats, also produce more catkins, and the seeds are more likely to germinate. Dwarf Birch is a really nice one to do this on because you can bend over and you can count the catkins. It's not like a tree where you have to try and struggle. And so if we project it into the future, we can see that Dwarf Birch is going to seriously decline, and under the worst situations, less than 1% of its habitat left. So goodbye Dwarf Birch in the UK. But is there anything we can do about it in the UK? Even though these species might be really common in other countries, we really, really worry about them. So what could we do to try and protect Dwarf Birch populations? So this is a figure that we put together, but it was adapted from Christian Rellstab's paper, I think in 2015, which we thought was super cool. And I'm just going to talk you through this really, really quickly. Each population here, each circle here is a population. And this is a genome environment association. So the dashed line is the theoretical optimum. And this is so we've got the temperature along the bottom, we've got the allele frequencies up the y-axis there. And so what we can see is we have a group of SNPs here. This is made up of about 17 SNPs that are really highly associated with annual mean temperature. And what we're taking is the distance of each of these populations to that optimum line is in some ways a measure of maladaptation. And I've been talking to people here at this conference in particular about what we should call this. We called it C-RONA, which is the current risk of non-adaptiveness, similar to what Christian called it, but maybe genomic offset or genetic offset, or there's lots of other things we could call it. We need to come up with a catchier name. And basically, we measured for each population how far they are from this optimum. And if you're a long way, then potentially you're maladapted. What does this mean? This gives you two options. One, you could do assisted migration. You could take that population and you could move it in this place to somewhere cooler where it would be happier. But perhaps that's not particularly feasible. The other thing you could do is assisted gene flow. You could introduce diversity, and you can use this graph to identify a group of suitable populations. You could introduce this diversity to your focal population to try and rescue it. And then, of course, you can extend all of this under climate change to more severe scenarios. But, of course, as you start projecting into the future, your uncertainties get bigger and bigger. So I'm just going to talk about adapting to current climate. And why is this interesting? Because in Scotland, a lot of our collaborators are collecting a lot of Dwarf Birch seeds from various populations, and they want to start putting them out. They want to start restoring. So it's going to happen anyway. So can we use genomics to try and guide that in the best way possible? So what does it look like? Lots of people have said assisted gene flow might be a nice idea. Maybe we should just start moving things from further south, further north. But this is what it would look like in Scotland. Each of these, this one is annual mean temperature, and this is mean diurnal range. And a blue population here is one that we think is already adapted. We can't find diversity elsewhere in Scotland that we think would be better suited to that location. So they're sort of the winning populations. And as you can see, they are mostly in the middle of Scotland, in the core of the range. And then the red populations are one that potentially need a donation of diversity. And the lines show you where we would suggest moving those populations to. So to be clear, I'm not saying let's go and do this straight away. Let's test, let's move some of these plants and let's see how they perform. But in a situation where it's Dwarf Birch and you don't have common garden experiments and you don't have nice setups because it's just a wee tree, this is what we would propose could be the beginnings of a strategy for the future. I'm just going to mention two other really brief things. I've talked to quite a lot of people over these last 2 days about the different signals you get from neutral and adaptive variation. And so what you can see here is the yellow bars are, if you like, genetic uniqueness or branch length, and the yellow are for neutral and the blue are for adaptive. So I mentioned these southern populations here. These are the only English populations, and they are quite rightly, or perhaps been a focus of conservation. And we find they have extremely high neutral diversity. So if we'd just done microsatellites and we'd just done neutral diversity, we'd say, hey, these things are so unique, these should be top priority for conservation. But actually, we find that is almost completely due to genetic drift, and they really don't contain much more adaptive diversity than any of the others. And that adaptive diversity is probably maladaptive anyway, because they're all subject to such extreme genetic drift. So it just goes to show if you look at neutral and adaptive, you get very strongly differing signals. And so we would say we need to look at both. And then the last thing, Christian showed this graph earlier, and this is more just to put it out there for anyone doing similar kinds of study. We found a really high association between the number of genome environment associations and the corresponding environmental variable. So for variables that are really important in our maxent models, in our species distribution models, so let's say temperature, we found a really high number of SNPs associated with those variables. So we find a really high relationship. Interestingly, Christian found completely the opposite, no relationship. So we'd really put it out there for anyone doing this kind of things to see if you find a consistent relationship between your species distribution model variables and your genome environment associations. So both of these things, all this stuff's published in these two papers. I just want to get 30 seconds in for one last quick mention. We're now working on what's called the tree against hunger, which anyone astute will realize it's not a tree, it's a banana. But we're now applying this stuff in Ethiopia for this super diverse food security crop. And we're trying to use this together with an app to be able to help farmers know which genotypes they should plant. So a bit like assisted gene flow, but for farmers to help them adapt to climate change in Ethiopia. Just to acknowledge all our brilliant collaborators in Ethiopia and in the UK and all the people that contributed to this. And there's the summary. Thanks very much for listening to me. Thanks.
Are there questions to James? Yes, please.
Well, thank you, James. I wondered if you would agree that, the diversity pattern that you showed with high diversity, no, not the diversity, the differentiation it was, the very small populations, you had a high diversity, high genetic differentiation. So would you say that kind of intermediate-sized populations would be in depth for extinction, genetic extinction?
Yes, you're probably right. One of the problems with them all being so long-lived and lovely is that you get this lag that you're waiting quite a long time. But I guess what I would say is that my preconceptions were that it would just get worse as you got smaller. But we found such a stronger signal, fine, and then only when we got to really small sizes, in our case, less than 10, did we see that signal. I guess the message is just that trees are potentially a bit more resilient than we thought. You can get quite small. Of course, I agree with you, if you're less than 50 perhaps, or less than 100, you still might be storing up those problems for the future, and that's also not ideal. But in our case, it was only really apparent until we got down to really small numbers, which for a lot of rare species is nice, yeah.
Hi, James. Thanks for the great study. Really like how you combine multiple markers, a lot of different methods. My question is about the recommendations for assisted gene flow. I saw the paper and I took a close look at the plots that you showed, and it looks like which populations pop out as blue versus red depend very much on the environmental variable you're looking at, even just for the two you showed. But I assume, when you start to look at more environmental variables, you get very different gene flow recommendations. So can you synthesize many environmental variables into a few? And or is that, what are some of the downsides of that kind of recommendation?
Yeah. So you're absolutely right. So what we did do is we synthesized across all the SNPs associated with one variable, so let's say annual mean temperature, and we produced a map. But the really logical, like the painfully logically brilliant next thing to do would then be to synthesize across all those environmental variables. So you just produce one map for the practitioners. And that's something we'd 100% like to do. We think we know how to do it, but eventually you have to draw a line and publish it and then put all the next ideas into the next paper, hopefully. But you're absolutely right. That's what conservation practitioners need. And it's 100% possible to do. It's just a trick of some of the variables. It's lots of arrows going in lots of places. And some variables, you've got lots of SNPs associated. So do you say that because you've got 200 SNPs associated with annual mean temperature, that you should prioritize how your gene flow fixes that? So you're right. It's just working out how to balance complexity of recommendation with something that's true to the actual system. Yeah.
One more question.
Sylvie Muratorio in her France. And to go further on these assisted gene flow things, don't you think that it will be more wise or secure or whatever to test first that difference in frequencies really translate into difference of fitness before, going into these practice, changing practice? I mean...
Yeah, so absolutely. One thing that was interesting is our metric of maladaptation, that C-RONA, that how far the population is from the optimum allele frequency, that was also very highly correlated with our fitness proxies, which was catkin counts or germination. So we like you said, we think we're right, but I guess we all kind of feel this. What we're talking about is potentially rearranging the diversity of trees across the whole of Europe or North America, and these things have spent a couple of million years getting to how they are now, so we should kind of be really sure before we go mess with them. But clock's ticking, so eventually you have to make a decision. But you're right, we should check that, and we'd like to, but we think we're right at the moment.